Welcome back, everyone. Dr. Linda here. Okay, in this upcoming video, we'll talk quite a bit about the cell membrane. So let's take a look. Now, the cell wall or the cell membrane, we consider it to be a phospholipid bilayer. It is a flexible but a sturdy barrier that surrounds the cytoplasm of the cell. Now, let me show you. If we take, let's see if I could draw this here. So simply speaking, let's say here is the phospholipid bilayer, and let's say right in the middle, that is the nucleus. We're going to take this section of the cell membrane. We're going to take it out a little bit and show you what it's made up of. But it is a membrane that's made up of 50%, about 50% fat and about 50% protein, or 50% lipids and 50% protein. The proteins are the gatekeeper of the cell membrane, right? It's responsible for what enters the cell and what exits the cell. We say it's a phospholipid bilayer, and they're held together by hydrogen bonds, and hydrogen bonds can be made or broken or damaged. And the proteins, once again, these are the gatekeepers. But let's see if I could draw this for you. So let's say we take a portion of that cell membrane. I'm making it larger. Okay, so this portion right here, we're going to do here. And outside of the cell, there is H2O, there's water. And inside of the cell, there's H2O, right? There's water, extracellularly and intracellularly. So on the outside of the cell, we're going to have these spheres that are hydrophilic. And on the inside, closer to the intracellular portion or the cytosol, we're going to have these spheres that are hydrophilic. Those are phosphates. And then on the inside, let's make this blue, we're going to have these lipids. And I'm making one lipid straight and then the other one bent. Straight and bent. And they're going to be on both sides, straight and bent. Straight and bent, coming off of the phosphate, straight and bent. So the reason why I'm making one straight is because it's saturated and the other one bent is unsaturated. So the saturated portion gives the cell membrane its sturdiness, and the unsaturated one that's bent like an elbow would give it its flexibility. Okay, and cells need to be strong and sturdy, but they also need to have fluidity or flexibility. So this is called the phospholipid bilayer, and it makes it selectively permeable. Now, the plasma membrane separates what's inside from what's outside, right? Because the conditions are very, very different. What's inside and outside is very different. The pH is different. What enzymes are found within need to be there. What's outside needs to remain outside. So the plasma membrane serves a very important function. It helps to regulate the exchange of what's permitted in and what's exits. For example, glucose, right? We just don't want glucose passively entering the cell because glucose in high concentrations is toxic. So we have to be careful. It's also involved in eliminating wastes. Okay, so it's involved in the regulation and exchange of substances. And it's also very sensitive to pH. Remember, the pH scale goes from 0 to 14. And right in the center is 7. That's neutral. Anything from 0 to 6.999 is an acid, and everything above 7 to 14 is an alkaline or a base. If the cell or parts of the cell or organelles become too acidic, it can destroy it. Okay. So here we have this phospholipid bilayer. We say it's a lipid bilayer, but I like to say it's a phospholipid bilayer. And it's back-to-back layers of different types of lipids. We see that there's also cholesterol and glycolipids that are scattered throughout. So we have some glycolipids here. We have cholesterol here. And notice here is the phospholipids, right? This is one layer of all of those spheres. And then here's the other layer of the spheres. And then in the middle, we see a saturated and unsaturated fat, okay? So it's a phospholipid bilayer, and we said that it's made up of fat, and it's also made up of proteins. So we see that we have some peripheral proteins and integral proteins embedded within the cell membrane. We'll talk about what their functions are in just a little bit. So we have a phospholipid bilayer, and we could see we have on this side is extracellular, that's outside of the cell. And on this side is the cytosol, that's within the cell. And the purpose of this phospholipid bilayer is to keep what's out, and to keep what's in. And then these proteins, whether they're peripheral proteins or integral proteins, are going to allow the exchange of substances in and out. Okay, these proteins, especially the integral proteins, they can act as channels, like a sodium channel, potassium channel. And we'll talk about proteins and how they can even carry things in and out of the cell, like glucose. They have glucose carrier molecules that can do that. We know that outside of a cell, we have more sodium and we have more oxygen. And we know that, and what we'll talk about in terms of diffusion, is that things go from an area of higher concentration to lesser concentration. So sodium wants to find its way from out to in. Oxygen wants to find the way from out to in. Okay. And then we'll talk about, well, after the sodium goes in, how do we get it out? Because sodium, being that its higher concentration is extracellular, things tend to go from higher concentration to lesser. We say it's following its concentration gradient. And things also go from, let's say, where it may be more positively concentrated to negative, right? These opposite, this opposite of positives and negatives always draw to one another. If something is more negative, it moves to where it's positive. If something's positive, it moves to where it's more negative. Okay, let's see what else we have here. So that is your lipid bilayers right there. And then there's your glycolipids, there's your cholesterol. The phospholipid portion, the water and glucose cannot cross the hydrophobic lipid portion, and it shouldn't, right? Because the internal and the external environments are very different. So it allows for this selective permeability. Now, fat-soluble substances, they can pass easily from the blood into the cell, and that helps explain why alcohol can pass quickly, right? And it can be absorbed from the bloodstream right into the neuron or right into brain cells pretty fast. And that's why we check neurology, finger to nose or heel to toe when you're walking to see that alcohol get into the cerebellum and your balance and equilibrium is shot and your fine coordinative skills are shot. Okay, the cholesterol that's within the cell membrane, that makes up about 20%. And, this brings up an interesting topic about, well, if cholesterol makes up 20% of the cell, then what happens to cells if someone's on an anti-lipidemic or anti-cholesterol medication? It does have impact on the integrity of all cells. We start to see an impact, especially the neurology. We see that one of the black box warnings on some of the anti-lipidemic meds, these statins, is cognitive impairment, because now the cell membranes are being impaired. So dementia-like symptoms or cognitive decline are one of the black box warnings on like simvastatin, Crestor, Lipitor. We have to be careful. And in another video, we talked about some of the drug-induced nutrient depletions with cholesterol medications, especially the CoQ10. Okay, so make sure you review the cholesterol pathway and how the statin does block HMG-CoA reductase, and it does downregulate the amount of cholesterol being produced. But at the same time, it will impair CoQ10. And if you're impaired of coenzyme Q10, what happens to the electron transport chain, which takes place within the mitochondria that we'll talk about when we do the organelles? The Krebs cycle and the electron transport chain both take place in the mitochondria, and they're responsible for ATP production. So first you have glycolysis, which is breaking down glucose into pyruvate. And if oxygen is available, pyruvate can enter the mitochondria. And that's where you have the Krebs cycle or the citric acid cycle. And you start to make these coenzymes. You do make a little bit of ATP, but you make some coenzymes, NAD and FAD. And NAD and FAD are utilized in the electron transport chain to really make lots of energy, or ATP, lots of it. And it's dependent on iron, and it's dependent on CoQ10. And if you're impaired or deficient in coenzyme Q10, then ATP production becomes impaired. Something to keep in mind. Okay. So cholesterol is really important. It's the precursor to testosterone and estrogen and progesterone. And that's why we see when cholesterol medications are used, it doesn't pair with some of the sex hormones. There's cholesterol. Okay, so the plasma membrane, it's flexible, yet it's sturdy barrier that surrounds and contains the cytoplasm of the cell. There are two types of membrane proteins. There are integral proteins and peripheral proteins. Now, when we look at them, you'll see the integral proteins, they're like transmembrane, they pass right through, whereas the peripheral proteins hang around the periphery of the cell membrane. But the integral runs either run into the membrane or go completely through, and they're responsible for making up many of the channels. So if we look here at what some of the functions of membrane proteins are, we'll see that they do make up channels, especially the integral runs. The integral proteins, they form a pore or a channel or a doorway or a window that can allow certain ions to flow and get across the membrane. Then we have carrier proteins. Again, these are integral proteins. Carrier proteins are like transport proteins. They can transport something across the cell membrane surface, especially something that maybe their molecular weight is too heavy. We don't want glucose traveling into the cell membrane unchecked, so it uses a protein carrier to bring it in. Receptors also integral proteins. They recognize specific ligands. You're going to see that term ligands frequently, and it alters the cell function. So ADH, antidiuretic hormone, can bind to a very specific receptor in the kidneys, and it changes the water permeability, right? Antidiuretic. It's not a diuretic. It's not going to make a person urinate. It's going to allow the person to hold on to urine. Enzymes, these are integral or peripheral proteins, and these are designed to kind of speed up the reaction time, okay? They catalyze reactions inside and outside of the cell, right? Enzymes speed up reaction time. We have digestive enzymes in our stomach, and they speed up the rate at which it can break down proteins and carbohydrates and fats, right? You can put a burger on a barbecue, but the temperature of the barbecue could be 400, 500 degrees, and it denatures and breaks down the protein in the meat. But our body temperature is 99, 98, 100 degrees. It doesn't get 400. So how does it break that down? It uses enzymes to accelerate the process. We have linker proteins. These are integral and peripheral protein. and they anchor filaments inside and outside the plasma membrane, providing structural stability and shape of the cell. Okay, and then cell identity markers. These cell identity markers, these are glycoproteins that we saw that hang out along the cell membrane. And this is what helps distinguish your cells from someone else's cells. Okay, it's an important class of certain markers that are called MHC, major histocompatibility. It tells the body, hey, I'm a kidney cell, or hey, I'm a neuron, or I'm a liver cell and I'm a stomach cell. All right, there are cell identity markers. It's the difference between someone who's blood type A versus someone that's blood type B. Okay, so membrane, we said this already. There's nothing new here. We know the importance of cholesterol also serves to stabilize the cell membrane and to reduce some membrane fluidity. It makes up about 20% of the cell membrane. The plasma membrane, these are selectively permeable. The lipid bilayer is always permeable to small, nonpolar, and uncharged molecules. The transmembrane proteins act as a channel or a transporter to increase permeability, right? Those proteins, the transmembrane or those integral proteins, can act as channels to transport things in and out of the cell. And then we have macromolecules are only able to pass through the plasma membrane by vesicular transport, meaning a protein can be packaged in a vesicle. And when I go over the Golgi complex or the Golgi apparatus, you'll be able to see these vesicular transport take place, how this vesicle can have a protein, it can move towards the cell membrane wall, open up and release the contents outside of the cell. We call it exocytosis, or it can actually bring something into the cell, pinocytosis or endocytosis. And a protein, this vesicular transport can transport this protein to the cell membrane, and the proteins are used to recycle the cell membrane. It's not being shipped out of the cell, it's being used to recycle some of the damaged proteins. You heard me use the word concentration gradient before. The concentration gradient is the difference in the concentration of certain chemicals between one side of the plasma membrane and the other. What do we have more of extracellularly, and what do we have more of intracellularly? An electrical gradient is the difference of the concentration between one side of the membrane and the other. Okay, and sometimes it's made up of, well, where's there more sodium? Where's there more potassium? Where's there more chloride? Where's there more protein? That changes the electrical gradient. So together, we call this an electrochemical gradient, where things go from an area of higher concentration to lesser concentration, and things go from where they're positive to negative or negative to more positive. Opposites attract. Okay. Let's look at transport across that plasma membrane. So we have passive transport and active transport. When we look at, let me just move this out of the way so I can see it a little bit better. When we look at passive diffusion, this is simple diffusion, facilitated diffusion, and osmosis. Osmosis involves water, and water goes from an area of higher concentration to lesser concentration as well. Simple diffusion is really things going from an area of higher concentration to less, like sodium going from outside of the cell to inside of the cell, or potassium going from inside of the cell to outside of the cell. But facilitated diffusion, facilitated means it needs a help. Can't do it by itself. It needs some assistance. We'll talk about active processes, primary and secondary transport. These things typically require some form of energy. You can click on this link on your own time to watch a quick video showing you transport across a plasma membrane. So membranes can maintain the difference in their concentration gradient of substances inside versus outside, or intracellularly versus extracellularly. But there's more oxygen and sodium outside of a cell membrane, and there's more carbon dioxide and potassium inside of the cell membrane. So these things will want to move in opposite directions. The membrane can maintain a difference in their charge of ions between what's in and what's outside. Okay. The substances move down their concentration gradient, and they always move towards oppositely charged areas. So this is what I said earlier, where negatives want to move to positives and positives want to move to negatives. So sodium, which is in a higher concentration outside of the cell, will want to move in, and potassium, which is a higher concentration within the cell, will want to go out. Sodium and potassium, they're antagonists. They usually move in opposite directions of one another. We know that if someone has too much sodium and not enough potassium, their blood pressure can go up. We know if they increase the potassium in relationship to sodium, blood pressure can go down. We know oxygen, think of my nose and my head here as a cell. Oxygen is higher out here. Now I breathe it in. And then I have to blow out the CO2, the exhaust from the cell. Okay. In terms of passive transport, let's look at simple diffusion. So if we look here at this test tube, right, here's water, and someone put this blue dye in here. So it was highly concentrated here. And then what happened? Things moved from a higher concentration to lesser, so it starts to diffuse. It goes down its concentration gradient from area of higher to less until eventually equilibrium is met. That's pretty cool, right? In facilitated diffusion, this is now where it's not moving without any assistance. It needs the help of something to travel through the cell membrane wall. So one of the things that it uses are the channels, the proteins. It can use a channel-mediated facilitated diffusion, or it uses transport protein, which is carrier-mediated facilitated diffusion. And you've heard of calcium channels and sodium channels. You've heard of insulin being involved in trying to move, to activate a transporter within the cell called a GLUT4 transporter. Insulin moves that GLUT4 transporter to the cell membrane, and that GLUT4 transporter opens up and allows glucose into the cell. That's a carrier or a transport protein. So here in this picture, we see that here we have a high concentration, and it goes from an area of high concentration to less concentration. So here we see simple diffusion. Certain things are small enough to passively diffuse through that phospholipid bilayer. And now here we have facilitated. This is using a channel. And here we have carrier mediated. This is using a transport protein. This would be like moving glucose in. Okay. Again, extracellular, cytosol, intracellular, going from outside to inside. So here we have passive transport. This is a channel-mediated facilitated diffusion. And this looks like we have potassium that looks like it is moving through this channel. Okay, we see that there have gates. Gates can open and close, nothing other than really a doorway allowing things in and out. Here we have a transport protein or carrier-mediated diffusion. So here's glucose, and glucose is going to use this glucose carrier protein to allow it to enter the cell. It can't passively diffuse by itself, so it's using help. It weighs too much to get in by itself, so it's using a facilitator. Let's look at osmosis. So osmosis is kind of like diffusion, but diffusion is using like sodium, potassium. It's using these ions and these elements. But here we're using, osmosis refers purely to water, nothing else. And we know water will find its way into any hole in a home, and that's how you get these leaks with these heavy rains. It goes from an area of higher concentration to lesser. So right here, we see in this tube, it's filled with water, and the water level looks the same. And there's this semi-permeable membrane right here in the center. But on the left side, we have purely water, nothing but H2O. And on the other side, we have water and we have some minerals. Now, water always goes from an area of higher concentration to less. And water likes to follow minerals. Okay, so the water is going to move where it's higher concentrated on the left-hand side, and it's going to be pulled in to the right side of the membrane, which raises that level. You see that? Okay, that's an important concept. So let's relate it to this first. Let's look at a red blood cell that is put into a type of solute. And it just so happens that here, isotonic means that the concentration of water and solutes is the same outside as it is inside. So things are moving equally in and out, in and out. So the red blood cell doesn't have opportunity to increase in size or decrease. It maintains it. But here, we have this cell put in a hypotonic solution. So there are more solutes inside this red blood cell than outside. Outside has primarily water. Outside is primarily water. So water is going to go from where there's a higher concentration to where there's a lesser concentration. So it's going to be rushing into that red blood cell, and the red blood cell increases in size, and then it bursts. They call it hemolysis or hemolysis. In this instance, this red blood cell is put into a hypertonic solution. So yes, there's water here, but there are more solutes in this water in relationship to what we have inside of the cell. So the water is going to go from where there's a higher concentration, in this instance would be inside the red blood cell. It's going to go to where the minerals are. Water always follows the minerals, and it's pulled outside of the red blood cell into the water, and the red blood cell shrinks. That's called crenation. Okay, so osmosis is the force that attracts water into a system. And we know that high protein diets tend to put a little bit more strain on kidney. So the dehydration is going to force kidneys to work a little bit harder to clear toxins from the blood. The kidneys filter blood, but the kidneys are also responsible for regulating blood pressure due to the renin-angiotensin system. The kidneys produce renin, and renin is one of those hormones that's really involved in converting angiotensin 1 into in converting angiotensinogen, which is produced by the liver, into angiotensin 1. Okay. If the body also perceives that oxygen levels are too low, then the kidneys also produce erythropoietin, EPO. And that hormone drives into the bone marrow and tells the bone marrow to push out more red blood cells. And the kidneys are also involved in producing vitamin D. So kidneys are really, really important. We don't want the kidneys to malfunction. When there's an increase in protein, that leads to the buildup of nitrogen in the blood. Nitrogen ends up in the kidneys in the form of urea. And urea has to be cleansed from the blood. It goes into the urine. And now the body's going to increase urine formation, and you're going to clear out these toxins from the body. And at the same time that you're clearing out the toxins, you're losing water, you tend to become dehydrated, and that could put a lot of strain on the kidneys. So hydration is really, really important. General rule of thumb is, you take your body weight, you cut in half and drink that in ounces. Okay, active transport is different than passive, right? Passive didn't require energy, but active transport is like swimming against the tide. It is walking against the winds, right? It requires more energy to do that. So what we know happens and takes place, if you look at the left-hand side, outside of the cell, extracellularly, we have more sodium outside. So sodium wants to go from an area of higher concentration to lesser. But there's also more potassium inside the cell, and it wants to go from an area of higher concentration to less. So now the question is, what happens with this sodium potassium pump that we've heard so much about and read much about? So how do we get the sodium that's now currently inside of the cell out? And how do we get the potassium that's now out of the cell back in to maintain this membrane potential so that we have a particular charge outside and a certain charge inside? So what happens is this sodium potassium pump, where you see in number one, it's taking 3 sodiums and trying to push it out. And at the same time, it's going to bring in 2 potassium. Okay. This is what happens. So you have 3 sodiums going out, and then we have 2 potassiums coming back in. 3 potassiums going out, 2 potassiums coming back in. Okay, so this is now saying, all right, we're going against the concentration gradient, and you see that it takes ATP in order to do that. Going from sodium coming in and potassium going out doesn't take any energy, but to restore it back to its membrane potential, we got to get sodium back out and we got to get potassium back in. And it does so by pumping 3 sodiums out and bringing 2 potassiums back in. Now, secondary transport, this is where, if you look, you'll see that we have sodium coming in, but look, calcium is sneaking out. We got sodium coming in, and we got hydrogen sneaking out through that same door. Okay, so it's like saying, hey, quick, the channel's open, let's get out. All right, that's called antiporters, where things are going in opposite directions. Now here, when sodium comes in, glucose can sneak in with it. And that's what makes soda and a lot of beverages that are high in glucose that do have sodium really bad for us, because they do raise blood sugar and they do raise blood pressure at the same time. You have sodium and sugar coming in, not a great combination. Okay, these are called symporters, where 2 things are coming in. Antiporters, one's going in, one's going out. Symporters, 2 things coming in at the exact same time. You can get protein and amino acids coming in with salt. And that's why when you have a steak, innately people go, it needs a little flavor, and you add a little salt to it. And with the salt or with the sodium, you can absorb a lot of the protein with it. Let's look at these pumps in relationship to this drug. It's also an herb. It's a drug called digitalis, and it's designed to slow down the sodium pump, which lets more sodium accumulate in a heart muscle. Okay, it's less, there's less opportunity for sodium concentration gradient to cross the membrane. It's keeping it within the cell. It's not going to allow it back out. So that means that the sodium potassium antiporter is going to slow down, and it's going to help retain more calcium within the muscle cell. And calcium is involved in muscle contraction. So it's designed to strengthen the force of contraction. That's how digitalis works. In terms of active transport in vessels or vesicles, the term exocytosis and transcytosis comes into play, where exo is the word exit. It's when a membrane enclosed secretory vessel can move towards the cell membrane and fuse with it, and it opens up and releases its contents. Maybe it's a protein, maybe it's a hormone, maybe it's a neurotransmitter, maybe it's a digestive enzyme, but that's exocytosis. Transcytosis is a combination of endocytosis and exocytosis. Endocytosis means you're taking something into the cell. Exocytosis is used to remove substances outside the cell. Okay, so that's transcytosis. Okay, when we come back, we'll talk about more of the structures of the cell. We'll get outside of the cell membrane for a bit and talk about some of the other organelles.